All right, well, thank you very much for your patience, and we'll get started. Thanks, everybody, for both coming in person and to those of you who are online, which is quite a large group, by the way. So that's really exciting for the fall meeting of SWOG in sort of rainy and chilly Chicago. But we'll give you some updates on where things stand with the SWOG Myeloma Committee. And I'll also ask that our co-chair and our research and statistics lead be involved, and maybe you can quickly introduce yourselves.
I'm Antje Horing, statistician on the committee.
And I'm Sikandar Alawadi from Mayo, Florida. I'm one of the vice chairs for the committee.
And I think we have a chair reserved up here for Christine Yee, who is on the way. This is a quick overview of enrollment to SWOG studies, and you can see that over the last 3 years, we've had really wonderful enrollment ranging from about 250 to 350. We've been one of the more active committees within SWOG, which has been really great to see. And I think, as you'll see, we have ongoing as well as about to open studies that I think will continue that trend. So thanks to everybody who's been involved in concept development. Thanks to all of you here and at home who've been enrolling patients. And also just a reminder that there are areas in the portfolio where we do have space for studies. So if you do have an idea, especially if you're a young investigator, we'd really like to help you and foster your career and hopefully get your concept into a trial and make you famous. So please let us know, send us an email, join us on our monthly calls, come to the spring meeting, which I think will be in San Francisco. We'd really like for you to be involved. These are the ongoing studies which we'll go with first, and we've got S2209, which is for patients who are frail or intermediate fit with newly diagnosed myeloma. And for that, I'll turn it over to Dr. Alawadi.
Thank you, Dr. Olaski. Once again, welcome everybody for joining, those who are here in person and virtually. I should also add that those investigators who might be interested in floating any new concepts, please also consider young investigators have an excellent opportunity to go through SWOG's annual workshop. If you apply to that and get selected, that's a very nice pathway. So let's talk about S2209 first. This is a phase 3 randomized trial for frail or a subset of intermediate fit newly diagnosed myeloma patients, comparing different regimens that I'll go over. And I think it's quite remarkable that yesterday during our meeting is when we heard that we had our first enrollment. So kudos to Des Moines, Iowa Mercy Hospital for getting the first patient on. That was excellent. So this is as of just a couple of weeks ago. These are the sites that are approved to be undergoing the activation, about 276 sites. Now, we don't get to see on our end at SWOG how many sites are actually active, but these are sites that had applied to CTEP and have gotten approval to run the study through their system. So we know that at least it's active in a few, and the accrual happened. I also think it was remarkable that the first accrual happened in a smaller community site, because that is exactly who this study is geared towards. Also, something to highlight that this study is going to be open at several VAs across the country. That was another very strong push. Dr. Chauncey, Tom Chauncey, who's our VA champion, has worked very, very hard to get this lined up. So we are really looking forward to the VA contributing patients, which would, one, bring it closer to a real-world population. Two, it would also probably help mitigate some of the healthcare disparities where typically underrepresented populations do not get enrolled. So we're hoping that this study will be able to overcome some of those barriers. Kudos to the SWOG team and the VA team to make this happen. So the background, I'll very briefly go over this. We know that the frail and elderly patients are an underrepresented population, not only in our clinical trials, and typically in the US, we've not even had any clinical trials geared towards these patients previously. For newly diagnosed myeloma patients in this group, VRD Light has been a regimen that we've used very frequently, but at the same time, there has been daratumumab lendex, so DARA-RD, that has FDA approval in the frontline setting and has been studied in this patient population, and is also available, excellent regimen. But nothing has ever been done to compare these two regimens head-to-head to identify what might be the better option for the patients. And as I mentioned, we were really interested in designing something that was a real-world study. So this is the study schema. Patients who are either a subset of intermediate fit patients, I'll explain that, and also frail patients. They would be registered, risk stratification based on either the frailty index, intermediate or frail, FISH status for risk category, and pre-registration therapy, because the study actually allows up to 1 cycle of treatment for the patients. So they would be randomized to these 3 arms. Arm 1 is VRD Light, followed by LEN maintenance. Arm 2 is DRD, followed by LEN maintenance. And Arm 3 is DRD, followed by DARA-LEN maintenance. I'd like to point out that the way we assessed was that VRD Light is the way it is given or designed. Arm 2 is kind of the way real world ends up using DRD regimen, that after an X number of cycles, the DARA and DEX are typically stopped and only LEN maintenance is given to this patient population. And Arm 3 is what is the intended use of this DRD regimen in this population with the continued DARA-LEN as the maintenance. Patients get 9 cycles of induction, then maintenance. Maintenance continues till progression or any unexpected or unwanted side effects. But it's also important to note that the study actually has dual primaries, because it's almost like Anjay, our stats lead, explained yesterday nicely. It's almost like we are roping 2 randomized clinical trials into one, because it was hard to come up with the consensus whether the maintenance for DARA-LEN DEX should be LEN or DARA-LEN. So now there is a dual primary objective. The study is comparing Arm 1, VRD Light, to Arm 2, DRD LEN, with PFS as the primary endpoint. And then it's comparing Arm 1 to Arm 3 for an OS endpoint. Dual primary, either one of them comes positive, the study would be positive. But I can assure you there are all other sorts of inter-arm comparisons of PFS and OS that are planned as secondary objectives. So everything will be compared to everything eventually, but we had to come up with primary objective stats, numbers, assumptions. So that is how it is set up. So all the PFS OS comparisons, overall response rate, time to best response, safety, and these are all the main secondary objectives. We also have an additional objective for MRD assessment. MRD is being done in the study as standard of care. So we expect the patients to have the MRD testing done because it is widely available and approved. We will be comparing MRD conversion rates and also looking at sustained MRD negativity. MRD testing is recommended or asked to be done at the end of induction, 9 cycles, and then at the end of 1 year of maintenance. There are some banking objectives. Tissue is getting banked, and I'll touch upon that in a bit. The correlatives are being designed for that Dr. E is heading. And some imaging-related correlatives also based on the PET scan imaging that is being recommended to be done, and then those images will be captured. When we talk about as I mentioned, stratification factors, frail versus intermediate, high-risk FISH, for any of these 5 abnormalities, deletion 17p, translocation 1416, translocation 1420, 1q plus, or translocation 414. This study is for a total of 507 eligible patients, but accounting for some 10% ineligibles, which is standard. The total number would be about 564 patients accrued. We have primary and interim analyses planned. As I mentioned, the eligibility, patients should have measurable disease. If they have non-IgG myeloma, then the M-spike threshold is much lower. then urine M protein standard definition, and also light chain standard criteria. Patients should not have had prior treatment, but they are allowed to use emergency corticosteroids. Now, this is where all those tweaks that we have made to the study to make it more real world and conducive to patient enrollment. So corticosteroids, short courses allowed, radiation up until 3 days prior to treatment is allowed. Most of the protocols will limit it in 7 to 14 days prior to trial. And then up to 1 cycle of non-DARA containing regimen allowed. So patients can get that treatment with their local physician before getting enrolled in the study so that they are not able to delay their treatment too much. It is also extremely important to note that this is a portion which I highlighted last night in our kickoff call, because this is going to cause a little bit of confusion if we skim through it too fast. So I'll take an extra minute maybe to explain this portion. We have made the inclusion criteria much more, I would say, inclusive. So for the frail patients, the hemoglobin can be above 7 as against the historical above 8. Platelets can be above 50,000. And ANC can be above 0.75. Again, that is against a prior history of ANC of above 1. And we are allowing growth factor and transfusion support to be able to come into the study if the physician feels that the cytopenias are related to the disease. Bottom line is these are standard regimens. The patient's going to get these drugs, these regimens as standard of care. If they have myeloma, they need to be treated on the trial to get this comparison done. For those patients who are intermediate fit or qualifies intermediate fit on the calculator, by the way, this calculator is available. I have recommended even during our kickoff call, please add this to your favorites so you're able to calculate this quickly. We should be educating our research coordinators accordingly as well. For intermediate fit patients, the hemoglobin has to be between 7 or 8. Below 7 would be excluded, and above 8 would also be excluded. I'll explain that part. Platelets between 50 to 75, ANC between 0.75 to 1, or intermediate fit patients can be RISS 3, or their creatinine clearance can be below 30. Now, why have we said this? Because for a hemoglobin above 8, platelets above 75, ANC above 1, RISS 1, 2, and creatinine clearance above 30, intermediate fit patients, there is already an ECOG study ongoing. We did not want these studies to overlap with each other. So right now, at least, these are the criteria for intermediate fit. We'll see if in the future that ECOG study gets done. Well, maybe we'll have an opportunity to amend this protocol. But for now, intermediate fit, slight tight controls. I should also very quickly point out for frail patients, any degree of kidney dysfunction is allowed short of dialysis. As long as the patient is not on dialysis, creatinine clearance doesn't matter, they can go on the study. So hopefully these criteria will make going on to the study easier. PET scan at baseline, end of induction at 9 cycles, end of 1 year of maintenance, that is post cycle 22, and that at any time if there is concern of disease progression or clinically indicated. And these PET scan images will be captured centrally. Bone marrow aspirate, peripheral blood, and core biopsies are to be submitted at baseline, end of induction, and at end of 1 year of maintenance, or disease progression, whichever occurs earlier. The guidelines for exactly what kind of tubes, et cetera, is in the protocol. I should point out the study is not providing any kits, so the tubes, et cetera, needed would be institutional supplies. Standard SWOG specimen submission guidelines. We also have patient-reported outcomes that are already built into the trial. Dr. Christina Gowan is the lead for that portion. We have the EOR TCQL QC30, which is to be done at baseline at months 1, 3, 9, and 18 after randomization. And we also have the patient-reported outcome CTCAE criteria for fatigue, pain, and neuropathy, and they will be collected at baseline and then every single month when the patient is on treatment. Those 3 CTCAE criteria will be collected every month. And it was helpful for SWOG to provide the information that each of these assessments for quality of life should take 10 to 15 minutes. That is important for us to know and be able to educate our patients and staff. This is the contact information. As I said, the protocol is already ready. I can talk a little bit about this sub-study, I guess, about Dr. Calverley is here, David Calverley, who's leading this effort. We are planning to bring in this sub-study of patients for antithrombotic prophylaxis. The idea would be, historically, we've presented at this meeting that patients would be randomized to Xarelto versus low molecular weight heparin. We're not going that route anymore. There won't be an intervention. But all these patients, data will be collected about clots, bleeding, what kind of prophylaxis they're getting, et cetera. That amendment is in the works. Once that happens, some amount of data capture may be increased to be able to get that data. But the nice thing is that all these 564 patients will be part of this antithrombotic analysis. So at the moment, I'll maybe not go over these details because, as I mentioned, it is no longer an interventional study. It is only an observational study, which will be rolled out very soon with a subsequent amendment. By the way, this data for the thromboprophylaxis will be collected only for the first 9 months of the induction. That is our goal. We do have a focus on healthcare resource utilization and financial toxicity components. That will also be baked into subsequent amendments, but hopefully will be just based on a little bit extra data capture so that we can do those analyses at the end. Dr. Yee is planning the correlatives and also imaging endpoints. As I mentioned, Dr. Gavin is leading the work for patient-reported outcomes for quality of life, and Dr. Brea Leip at the back of the room is also one of my co-chairs. So we're very excited. We are glad the study is now finally active. Enrollment has started. We look forward to patients being enrolled on this soon. But I guess any questions, any thoughts from anyone? Anything we can provide clarification with? Maybe online also if there are any questions.
There's nothing on the chat.
Yes, that's absolutely. That's an excellent thought. Please, those who are online or logged into the Zoom, you're more than welcome to send your questions through the chat. We have that open. We'll be happy to read the questions and then respond to them right now. So other than that, I guess Dr. Manuel Gutara is also here. He's our SWOG Myeloma Committee DEI champion, and with him we have a plan to not only enhance but also monitor minority enrollment in this trial. And we have a plan of how we'll be assessing, setting up what we expect to be enrolled, and then at the interim analysis, we'll also try to look at the numbers of whether our enrollment is going on target or not. And if yes, great. If not, why not?
A couple of just rudimentary questions.
No, please.
You say any degree of renal insufficiency, is that creatinine clearance less than 60?
That's creatinine clearance even less than 15. As long as the patient is not on dialysis and they are frail, they can go on.
But what do you define as renal insufficiency?
Any creatinine clearance.
Of what? But they have to have renal insufficiency, right, to be in that one category?
No, they don't have to have. They could have normal kidney function, 60 to 90, 30 to 60, 15 to 30, less than 15. Any of that is allowed.
Okay, thank you.
For the frail patients. For the intermediate fit, they have to be only those are allowed whose creatinine clearance is less than 30, but they are not on dialysis. And that is because those who are creatinine clearance above 30 qualify for the ECOG trial, and we did not want the studies to compete with each other. So frail patients, irrespective of any degree of kidney dysfunction, can go on. And intermediate fit have to hit any one of those markers. It's not all of them, of either creatinine clearance less than 30, hemoglobin between 7 and 8, platelets between 50 and 75, ANC between 0.75 and 1, or RISS3. As long as intermediate fit hits one of these criteria, they can go on.
Okay, it seems like high calculus. I'm just looking at it from the side room. Other question I have for you is, in the time it took for the study to get up and running, it's too late now, but is there truly equipoise, I would say, in the academic community of RVD light versus DRD? Because I would say, when I refer, my patients, Dr. Lasky's old institution, they all get DRD.
Great. Excellent question. I think that's a very, very important question, spot on. So what we've also been doing is that while it has taken time to get to this point of this study getting activated, we've been in communication with pharma to, at least I've been looking at their market scan data, and it seems that while DRD has caught on after the Maya data got released, but for this particular population and even otherwise, VRD light is somewhere about a 40, 45-ish% of market share. DRD is about 40, 45-ish%, and then there's a sprinkling of Rev, Dex, Velcade, Dex, all different, Cyborg-D, all different kinds of things. So it seems that still it's up in the air because there is, if not in favor of one of them, it's a very equivalent kind of current usage in real world. So hopefully this will lend some further information about that. Appreciate your questions. Thank you.
Yeah, and also for this study, not just because of the efficacy we want to compare, but also the toxicity and patient-reported outcomes. Those are all equally important for patients.
And I think a great example of a study where we've got lots of questions we're trying to answer, which are being led by different investigators. So hopefully the results will be exciting. It's great to hear that we've got our first enrollee from Iowa, and it sounds like Dr. Ailawadi is going to keep a hawk eye on what's happening with the study. So let's move on to the next study for updates, which is the maintenance trial of DARA plus LEN versus LEN. And Dr. Amrita Krishnan is available, we hope, over Zoom. So I will turn it over to her, and I will serve as her Vanna White by advancing the slides.
Thank you, Vanna. I will start. Thank you, and I'm presenting on behalf of myself and Saurabh Chhabra, who is the co-chair from the BMT-CTN. If we can have the next slide, please. Just to remind people of the schema, this is a phase 3 randomized. There's 2 randomizations, initial randomizations post-autologous transplant, randomized to LEN versus LEN-DARA. And then the second randomization, which is the one that I'd say now approaching and have a fair number of patients on, is the MRD assessment at 2 years, where patients who are MRD negative are randomized to either stopping or continuing maintenance on their assigned arm. Next slide. Reminding people here, the primary objective is overall survival from that first randomization. Secondary objective, which is going to be the MRD negative rate between the treatment arms. And we are going to have some early reporting once we reach that in terms of that MRD reporting out. And the third thing I do want to sort of mention is that a very important part of the study, and especially when you think about maintenance, is quality of life. And Natalie Callender is leading that effort in terms of the QOL analysis, and we're almost completed accrual of the 250 patients who are part of that. And I also want to thank the SWOG Quality of Life Committee for all their help in making that possible. Next slide. So this trial has been many, many years. It started out actually as a different iteration with different agents, ultimately became its current form, and it was opened in 2019. As you can see, and we enrolled our first patient fairly quickly after opening. We added Canada in 2022, which was very exciting for us. And thank you again, SWOG Operations was a huge part of figuring out some logistics with drug delivery for that. We added another amendment for null antigen phenotype to try and also include more patients. That was revision 8. And then I will get to our second bullet here, the temporarily closed to accrual, which is kind of where we are, and explain that in more detail if we get to the next slide. So, as you said, we had robust accrual. We met our original accrual goals of 1,141 patients. Um, but as those patients reached that second randomization time point, the 2-year assessment, I don't know if it's showing on the slide, it has become clear that we needed to increase our accrual goals. And that really is based upon some MRD failure rates. If we can go to our next slide, you can see here that accrual was very robust and over our actual projections, which was amazing. And thank you to everyone in the room and online for that. Next slide. Then again, even by the time when we had to complete our original accrual and we had gone up to 23 patients per month. Next slide. And these are the centers again who were the lead accruers in this. Next slide. So here's the issue that I alluded to. So we reached our initial accrual, but this is something we couldn't anticipate till we had patients reaching that 2-year time point when we looked at MRD. And what we found is we had a pretty high failure rate of MRD. And so we weren't able to randomize patients because of that. And we did certain steps already a priori to try and take care of that. One is in an earlier amendment, we in fact made it mandatory that patients have a baseline MRD sample to be eligible. So that did help. And certainly with those changes and a lot of discussion with sites, again, sending an optimal specimen for MRD and the importance of that, we were able to decrease our MRD failure rate to below 20%. But there's still issues that are beyond our control in terms of sample quality, as you can see here, or polyclonal samples at presentation, which are now really about 15% of cases. So because of that, we really need to make sure we have adequate patients for that 2-year randomization. We made a decision after much discussion with the NCI, with our statisticians, to increase our accrual. So we submitted and approved an amendment, revision 8, to do that, as you can see here, increasing from 1,100 to 1,420 patients to, as you can see, account for that MRD issue. And we also clarified a definition of MRD negative. I think that was also a learning curve for us when we got to the 2-year time point to really be very constitutional about how we called it. And Dr. Chhabra and I actually review every MRD report together before we notify the center. And I also want to thank Dr. Jacobs, who helped us really educate us and helped us in terms of reviewing those reports. Next slide. So really the whole thing is the amendment is approved. Pending activation really is in regards to contracting. And we've been waiting now about 3 months with bated breath literally every week to hope that that contracting issue ultimately gets resolved so that we can reactivate. Some other things we've done in the interim to address certain very important questions that people ask is cytogenetics. Obviously, people have asked, are we being biased? Are people not enrolling patients of high-risk disease onto this? So we have a subgroup of champions, and I thank all of them who are listed here because we are literally looking at patient-level FISH reports and entering that data and adjudicating that. And we've had multiple meetings in terms of definitions as well as how to read those FISH reports and agree on that. So that is currently ongoing. Next slide. Some other sort of housekeeping things that we did as we learned. Fortunately, these are minor in terms of the MRD, but some sites a couple sites had called MRD by flow, and we had to re-educate that the only MRD that can be used for randomization is the one through adaptive. And another interesting thing is, not surprising, well, actually surprisingly, some patients, when they were told they were MRD negative and randomized to stop therapy, didn't want to stop therapy. So again, education that please, when you consent the patients, to remind them that this is one of the key parts of this trial. And then also because we want to make sure that we are able to call complete remission, which we can't unless we have a UPEP, and so that we're not underreporting. We also made that 24-month UPEP mandatory, and we don't release the MRD results sort of as a carrot till we know that a UPEP has been done. So as I said, we are just waiting for activation and when we can enroll again. Next slide. And again, I'd like to acknowledge everyone who's been part of these efforts and, thank you for your patience. As I said, once we open, we hope people will continue to support the trial, and hopefully if they do at the same levels, we will finish in a year. So I'm happy to answer any questions.
Okay, any questions from the audience here or those of you at home or at work on Zoom, please send us a message or question through chat, and Antje is monitoring that. And looks like there are none, so we will keep on moving. And the next study is an S2005 update, which is for newly diagnosed Waldenstrom macroglobulinemia patients. We've got Drs. Ailawadi and Bhutani as study chairs. Maybe I'll have Dr. Bhutani come up and give us an update since Dr. Ailawadi's already had his turn at the mic.
Thanks, Dr. Orlowski. So this is our trial S2005, which has gone through significant changes. Initially, this study was conceived as a comparison of ibrutinib venetoclax rituximab versus ibrutinib rituximab. And because of some reasons that I will bring up, this now has been modified to a phase 2 randomized trial of IR versus VR. And we are hoping to open the study, in the next, or reopen the study in the next month or two. So as I said, that this was initially a comparison of IR versus IVR, but as the study was completed and then opened, and as we had enrolled one patient on the study, we got a report from another center who had a phase 2 trial of similar combination showing that there were some arrhythmia-related deaths on a combination of IVR, and 2 cases of sudden cardiac deaths were seen. And because of that, we at that time decided to put the study on hold and make an amendment. So currently we are working, the amendment is making through CTEP right now. We are at last stages of those amendments and hopefully plan to open the study soon again. So this is just a brief background on venetoclax, why we are designing the study. This was a phase 2 trial done at Dana-Farber that evaluated single-agent venetoclax in relapsed refractory Waldenstrom's and showed an overall response rate of 85%. And a VGPR rate of around 19% and a major response rate of 81%. Overall, very well tolerated, with the main adverse event being neutropenia. There was no evidence of cardiac toxicity and 1 episode of TLS. As everybody knows, ibrutinib tends to be a challenge in some of these patients. Being older and having heart disease puts them at risk of arrhythmias. So, a significant number of these patients are unable to tolerate ibrutinib or BTK inhibitors very well. So that's why we want to, evaluate venetoclax in this setting to see if this would be more effective as well as possibly an easier and a safer regimen. So this is the new trial design. We are going to compare ibrutinib rituximab and venetoclax rituximab. Patients will be treated for a fixed duration of time, which is 24 months. So if patients have stable disease or better at the end of 24 cycles or then the patients will stop treatment and will be followed up for 5 years. If during that time, if they have evidence of disease progression, then they could go from IR to VR or VR to IR. and then again get treatment for 24 months. And if at that time they still have stable disease or better, then they will come off treatment and follow up for another 5 years. So the main reason for modification are safety reasons, and currently there is no efficacy data for VR in the newly diagnosed patients. We think that this study has the potential to generate good efficacy as well as safety data in this patient population. As well as ability to test patients for a limited duration of therapy. Currently, many patients are getting therapy for a very long time based on the current data with the ibrutinib as well as other BTK inhibitors. As well as we hope that this will generate some safety data to see if venetoclax could be safer from a cardiac standpoint as compared to the BTK inhibitors. So this is some of the points that I already discussed. So the treatment will be given for a fixed duration of time. The crossover that I already discussed. We have added some cardiac assessments at various time points to make sure we are assessing the cardiac safety. And the primary objective, which was complete remission in the past, has been changed to VGPR because we don't think many patients will achieve a complete remission because this is now only a doublet instead of a triplet therapy. These are the inclusion and exclusion criteria. The patients have to have an untreated Waldenstrom's that requires treatment now. The only treatment that is allowed is rituximab as long as the last dose was single agent rituximab as long as the last dose was 12 months or more prior to registration. Creatinine clearance of more than or equal to 30. These are the blood count requirements of platelet count of 50. Hemoglobin 8 and ANC of 1,000. There is venetoclax, as you guys know, has some interactions, and if patients are receiving a moderate or strong CYP3A inducer or moderate or strong CYP3A inhibitor or a PGP inhibitor within 7 days, those patients will not be able to enroll. But if the patient can be switched over to an alternative agent, then they can be enrolled as long as they don't have a major interaction with venetoclax. These are our new calculations for the statistical design. We think that the VGPR rate on the VR arm will be about 50% as compared to about 25% in the IR arm. And based on that, we have an calculated N of around 92 with 41 patients per arm, and the accrual rate of 2 to 3 patients per month. So happy to take any questions or comments. So the current status is that we are expecting a meeting with CTEP on Monday. And we hope that at that point, we are very close to then reopening the study. We will very much appreciate if everybody opens the study because we do want the study to enroll and hopefully be able to complete accrual in timely duration because that is going to be a sort of, our major sort of goal of doing the study. Yeah.
Yes, sorry, I had a question. Earlier this year, the German CLL group published in New England a 4-arm study using obinutuzumab, venetoclax, and ibrutinib. That was one of their arms. I didn't really see a lot of cardiac events in that arm when compared to the other ones. This is a different disease, was for CLL, but I was wondering if maybe there's something specific about Rituxan given in that combination that may have caused the side effects. And I know maybe a little late to give OV a consideration, but just a thought.
Yeah. So I think that's an excellent question. I can say that when the safety signal had come out, and that was from a different trial, as Dr. Patani clarified, quite a bit of a deep dive was done throughout because ibrutinib venetoclax is a combination that has been used in CLL and lymphomas and is not showing up any of that issues. So another quick analysis was done, very helpful from pharma standpoint also, that they helped look at their data because patient safety is paramount, obviously, for everybody. But it seems somehow that that signal was not there in any of the CLL or lymphoma studies. So that combination doesn't seem to be causing any issues. And I'll just clarify that the study at the other institution where those grade 5 events happened was ibrutinib venetoclax. They did not have the monoclonal antibody in that combination. It was a 2-drug combo. But because those 2-drug combination was a part of our triplet, so we immediately halted. Our patient, I should say, was able to just stay fine and had to come off the trial. We removed them from the study, but they did not have any adverse event of concern. So it seems somehow Waldenstrom has a different biology, which we have seen in other drugs also. For example, IMiDs are not easy to give in Waldenstrom. IMiDs are used routinely in myeloma and then, of course, also in CLL. So there seems to be something specific about the disease biology. For those grade 5 events, testing was done about IgM levels, viscosity, concern about light chain deposition, hard analysis, et cetera, et cetera. Somehow nothing came out. But excellent question, thanks.
And I think the other part of the equation there was that Waldenstrom generally is considered a fairly indolent disease, so your tolerance for grade 5 toxicities in a disease where people generally do pretty well for many, many years is much lower than if it were a disease where everybody had a 1-year overall survival. So I think that was the other part of the equation there. Okay, next are upcoming studies, which include S2213, and we'll ask Dr. Hagan to come up and tell us what's going on with that study, since he came a long way to the meeting today.
All right, thank you. So thank you, Bob, and everybody. So this is a study that we presented a couple of times now, and kind of pleased to say we are just about ready to activate. So we'll kind of go over the study and then obviously open up to any questions. So this is a randomized phase 3 trial, really asking a question we've been wanting to answer, I think, for many decades, and really, truly, what is the role of high-dose chemotherapy in autologous transplant and systemic light chain amyloidosis? Importantly, this is a SWOG-led study, but really has been an intergroup effort with ECOG, the Alliance. It's formally supported by the BMT-CTN, and really a lot of, I think, important input from many other stakeholders, even beyond that along the way. So just, again, in brief background, the only really truly randomized trial looking at this was done over 20 years, well, almost 20 years ago, by the IFM, where they compared high-dose chemo and transplant to oral melphalan. This is really an outdated study. Transplant-related mortality was high, and obviously, in current day, that's not the therapy we use in this disease anymore. So it's really, since that trial was completed, there's always been a lot of lingering questions, and really it didn't answer the definitive question we're trying to. I think importantly, transplant-related mortality has dramatically reduced, and I think that's an important message. And in most centers, it's less than 5%, and there's a CABMTR report that said you had to be an experienced center to have a low TRM, but in fact, there's many small case series that really show with proper patient selection, it's prohibitively, or it's not, it's an acceptable transplant-related mortality today. So, furthermore, induction therapy, meaning therapy before transplant, has also become the standard of care over the last 5 to 10 years, and we really know that along with the dose of melphalan you get at transplant, really dictates the long-term positive outcomes we see in these patients who get to transplant. And then I think also importantly, the fifth bullet point here is that practice patterns really are quite different. In the US, the data would support, although it's kind of a little hard to tease out, maybe about 1/3 of transplant-eligible patients receive an upfront transplant. In Europe, that number is much less. The large registry study, the Alchemy study, showed that was less than 5%. So again, we kind of speak with our actions, and the actions are very different around the world. And then I would just finish with, although previously challenging, more recently there's a bigger appetite for enrollment in patients in amyloid trials. We proved that here in our SWOG trial, looking at isatuximab in the relapse setting, and the fact that the Andromeda trial really enrolled much quicker than even anticipated. So I think it goes to show that some of the old kind of dogma, and I think probably incorrect stereotypes about amyloid in clinical trials really isn't holding true anymore. So hence this question. So this trial, again, is looking at optimal consolidation in these patients. The primary outcome is this modified progression-free survival, which is a composite endpoint, really looking at the most important outcomes in these patients, which is obviously overall survival, but also hematological progression and then organ progression, namely cardiac and renal progression, because we know that these are really the organs that drive poor outcomes in these patients. So that's the primary objective of the study. It will be to compare that between the two arms. There's a bunch of important secondary outcomes we're looking at. Quality of life will be critically important on this study. Overall survival, obviously the individual organ responses. We're going to be looking at minimal residual disease, kind of by standard flow-based assays in the bone marrow, but also I think very importantly on quite frequently corrected peripheral blood specimens to look at mass spec assays in the blood. We'll also be looking at responses, hematological, and then not transplant-related mortality, but treatment-related mortality. Because whether or not these patients, get an upfront transplant, unfortunately, just the mortality from the treatment itself is something of note. So these are all very important secondary outcomes. Here is our treatment schema, so depicted on the right. So patients, when they're eligible, which I'll show in the coming slides, they would go on through registration step 1, at which point everybody would receive standard induction therapy with dara-VCD or dara-CyBorD for 3 cycles. After which, if still eligible, patients are randomized to go on to high-dose chemotherapy and stem cell transplant, or to finish the standard 6 cycles of dara-VCD. Following which all patients will go on to 18 months of maintenance daratumumab. So here's the schema. And then in terms of, I think, key eligibility, so these are driven mainly around transplant eligibility and some standard clinical trial eligibility. I think importantly, age has to be less than 70, and that's kind of based on data around the country that those patients aren't really getting transplant and are frequently excluded in many transplant eligibility criteria that are set by some of the leading groups in amyloid. They do have to have a measurable difference in their free light chain, and that's because we have to be able to measure responses, so that's got to be greater than 2, and that's because we have validated response criteria in those patients. And then the standard non-severe cardiac markers, so the NT-proBNP and TNT. I think importantly, we do have built into the protocol the ability to measure that with both BNP and NT-proBNP, as well as troponin T and I, because those are frequently checked differently at different institutions. So we have a way to kind of cross-validate those markers. And actually, it's another, I think, secondary question we'll be actually asking is there a correlation between those in these 2 different approaches? Creatinine clearance has to be greater than 30, and then I think importantly, these patients are without at least 1 cycle of plasma cell-directed therapy before enrollment. And that's, again, similar to what we've talked about on previous studies here today. That's to capture those patients that are started on therapy in the community and then are referred later for potential trial enrollment. So after they undergo their 3 cycles of induction, they have to still be eligible for high-dose chemotherapy. So again, these are essentially all the same criteria I just mentioned. The only additional one highlighted here in red is that they have to have at least a partial response to their induction therapy. So at that point, patients would be eligible for randomization, and then if those who are randomized to high-dose chemotherapy have to collect enough stem cells to actually move forward with the transplant to remain eligible at that point. So in terms of our methodology, as I said, the primary outcome is the modified progression-free survival based on kind of available data on the Andromeda study. We've estimated that to be about 31 months in the non-transplant arm. We're looking to see about an 18-month improvement up to 50 months in the transplant arm. So this is, we would need to actually randomize 143 patients per arm to reach that endpoint. Importantly, the final analysis will be done after 192 events, but no later than 4 years after the last patient is enrolled on the trial. So due to obviously dropout, we're estimating that to be about 15%. That'll be a few patients with less than a PR, although again, kind of the data would support that's 5% at tops. And then we're going to get some dropout, obviously, due to toxicity and then ineligibility at the time of randomization. So ultimately, we'll need 159 participants enrolled in each arm for the trial to meet its endpoint with 143 eligible patients for randomization. Important stratification factors are the European modification, the Mayo 2004 staging, which really captures cardiac involvement, response after their induction, so PR versus a VGPR better, and then presence of 1114 translocation, which can impact outcome with high-dose chemotherapy, melphalan namely. So, and then in terms of kind of the translational endpoints, there is many, but I think really importantly, we're going to be looking at, again, minimal residual disease in the bone marrow. This will be done by next-gen flow through the help of the Mayo and Rochester, as opposed to kind of the clonoSEQ we're seeing on other tests for a variety of reasons. So in this, the primary will actually be post-consolidation we'll be looking. And then secondary and exploratory endpoints, essentially we're going to be looking post-induction, post-consolidation, and 1 year post-consolidation, looking at the two different methodologies, so peripheral blood versus peripheral blood mass spec versus bone marrow flow, and then also trying to potentially correlate those two different methodologies with their primary endpoint of modified progression-free survival. So I think at the end of the day, this really is going to allow us to answer some important questions between the correlation of those tests and how they might individually impact both modified progression-free survival and outcomes in these patients. Also very important is that we have some integrated PROs built into the study. We're going to be utilizing the PROMIS 29+2 profile, which is essentially a slight modification of the PROMIS suited to amyloid patients. I would say this is a critically important part of this study because these two outcomes could very well be very similar and not different. And then the quality of life may actually dictate which patients you're taking forward in terms of the differences in toxicities, which we'll be assessing obviously individually on patients on trial. So this has kind of been our more recent timeline, and we have a few minor things, but we're hoping actually to activate the study this month. So this has obviously been a long time in the coming, but we're optimistic. And I would just say again for this group, and I said it earlier in the transplant meeting, there remains a lot of enthusiasm for this trial. This is a question we continue to see in our clinics. Patients continue to ask us, and we're hopeful through our effort here in SWOG we can definitively answer kind of the role of transplant in these patients. So with that, I will take any questions or any comments from the group or anybody online.
Okay, looks like we do not have any questions online and no questions here. So thank you for the excellent presentation. Yes, of course. I think she's next anyway, isn't she? Next is our high-risk myeloma concept. And we'll ask Dr. Abrea-Lightby to come up and give us an update on where this stands.
Thanks, everyone. So this is a trial we've been talking about for some time. This is a phase 3 study of the optimal induction and MRD-driven maintenance strategy for patients with newly diagnosed high-risk multiple myeloma. So we know that this disease remains incurable and that patients with high-risk disease in particular tend to do worse than other patients. So we still need to come up with the best strategies for treating these patients and prolonging their lives. So this trial, as it currently stands, we are looking at patients who are newly diagnosed with high-risk myeloma. They will be randomized to either RVD-DARA induction versus RV-DARA-TEC induction. They will get induction for 12 cycles. We will check MRD status by next-gen sequencing at 9 months and at 12 months. Patients who are negative at both of those time points will go on to a de-escalated maintenance approach with lenalidomide and bortezomib. Patients who are MRD positive at either of those time points will go on to a more intensified maintenance, either RV-DARA versus RV-DARA-TEC. So the primary objective for this study is overall survival between the two arms. We're going to look at a bunch of sub-studies looking at progression-free survival. We're going to look at toxicity rates. We're going to look at MRD status. And then we're also going to build in some quality of life analysis and some more molecular analysis. Patients must have newly diagnosed multiple myeloma. They must have high-risk disease, so this will be defined as RISS stage 3 disease or R2ISS high-risk disease based on the newest International Myeloma Working Group criteria. As our understanding of high-risk disease evolves, we anticipate that this eligibility criteria might change so that we can always make sure that we're capturing our high-risk patient populations. However, this trial is not for patients with plasma cell leukemia or amyloidosis or non-secretory disease. So basically, patients will be induced with RV-DARA or RV-DARA-TEC. They will get their maintenance after 12 cycles, which will be determined by whether or not they were able to achieve 2 MRD negative time points. So we are hoping to enroll 232 patients, and we've built in some dropout because we know that there's some issues with the MRD testing, so we've planned to over-enroll a bit so that we can achieve our endpoint. We expect that this trial will take about 8 years to complete. We also anticipate that as we attain greater understanding of how to dose some of our bispecifics, the dosing and the frequency of dosing for teclistamab over the lifespan of this trial might change as we understand that maybe we can give it less often. We are hoping to build those modifications in as needed. And then we're going to look at patient-reported outcomes and quality of life because in this patient population in particular, that is really critical. And I'm happy to take any questions.
All right, looks like, again, we don't have anybody at home with questions. Then we have other cooperative group studies that we're supporting, including the DETER study, for which we have a new champion, Andrew Cowan. Is Andrew on? It sounds like that'll be a no, so I'll go over this quickly. Whoops, not that quickly. As you remember, this is a randomized trial looking at lenalidomide and dexamethasone for 2 years versus daratumumab, lenalidomide and dexamethasone for 2 years for patients with high-risk smoldering myeloma that have to be within 12 months of their diagnosis. And then you have the option, if they have CR, PR, or stable disease, to continue therapy for 2 years, or off therapy, of course, if there is progression. This is the accrual summary so far, and SWOG is making a contribution, although there is still plenty of room for improvement there. So for those of you that do see smoldering patients, please try to either enroll them, or if you don't have the study open yet, there is time to do so, and would really appreciate your help to try to get this over the finish line. I think it's actually a pretty well-designed study because either of the arms should give you a longer progression to symptomatic disease than doing absolutely nothing, which would be the current standard of care. Next is the EQUATE trial, and we'll have Dr. Gowan come up and present since she's our champion for this study.
Welcome. Thank you so much. It's an honor to champion this trial. This is PI Shaji Kumar for ECOG trial EAA181. And so this is effective quadruplet utilization after treatment evaluation, a randomized phase 3 trial for newly diagnosed multiple myeloma not intended for early autologous transplantation. So a little background. We know that quadruplets are truly becoming standard of care for transplant-eligible newly diagnosed multiple myeloma. This is really from our Griffin trials, our master trials. We also are using triplets for standard of care for transplant ineligible, and it will remain to be seen who reigns supreme from S2209, a plug for this trial, which is very, very important. But there's still some unanswered questions out there, right? How much benefit is the additional fourth drug with respect to survival, which is the primary outcome of this trial? And does this benefit apply to all patients or only to those achieving deep response, not achieving deep response to those upfront triplets? So that's really what this trial is getting at. And so just to look at the schema, Newly diagnosed, again, are ISS stage 1, stage 2, not intended for early transplantation, and they can have received 1 cycle of therapy prior to enrollment. So this is really encouraging our community sites to participate. They are then randomized to daratumumab, Revlimid, or lenalidomide and dexamethasone times 9 cycles. MRD testing is then approached, and then they are randomized to DRD plus bortezomib, so that quadruplet, versus continuation of Dara-RD for additional 9 cycles. Maintenance is then continued, daratumumab, lenalidomide, until progression. Again, primary outcome is overall survival. Here is the progress so far. So we have 171 patients now on intervention. 27 of those are attributable to SWOG, so kudos to all those SWOG sites. There is more room for improvement, though. Lots in screening 35. We can certainly, hopefully, contribute more. You can see the target accruals is a little bit behind schedule. The targets are the dashes, the actual are the solid line below. So we really want to support our colleagues in ECOG and contribute to this really important trial. Kudos to those who are already doing so, Oregon, Yale, and University of Arizona. Thank you for your contribution. Thank you.
Very good. Thanks very much. S1702 is next, and I think I saw Terry Parker. There it is. Terry, you are. Okay, come on up, Terry. Dr. Parker will present where we are with this study, which was our first foray into relapsed AL amyloid, but perhaps not our last.
So a reminder of S1702, as it's been a minute. So we accrued our first patient back in 2018, and the accrual went quicker than expected. As you can see, it was closed in September of 2019. As a reminder, all patients with relapsed refractory AL amyloid who had received at least 1 prior therapy and had 1 organ involved received isatuximab for up to 24 cycles, so roughly 2 years. We presented patient characteristics and the initial safety data as an oral abstract at ISA back in 2020. And then the primary endpoint, which was overall hematological response, was presented at ASH in December of 2020. Currently, we just finalized the organ response data, and we just have 1 additional thing to look at with our cytogenetic data, and then the manual script will be ready for publication. So we're excited to get that out, hopefully in the coming months for everyone to see. And that's all I have, but happy to take any questions.
And
Thanks.
no questions from the audience, at least at home. Any questions from folks here? Okay. So hopefully we'll see an abstract of the final results soon, and then a manuscript. I think that's all that we have for you, and we do have a little bit of time if folks want to discuss any other topics or bring up items to put on the agenda for the fall meeting or for the next monthly call. If not, thanks everybody. Enjoy the rest of the SWOG meeting, and happy Halloween for the end of the month. Yeah, well, that's good. We have time. We still have things to talk about.